السلام عليكم. محاضرة بعنوان Genetics of Viruses لطلبة الدراسات العليا. Genetics of Animal Viruses. Genetic analysis is a powerful approach toward understanding the structure and function of the viral genome, its gene products, and their role in infection and disease. Viruses that have stable antigens on the surface, such as poliovirus, measles virus, can be controlled by vaccination. Other viruses that exist as many antigenic types, example rhinoviruses, or those can change frequently their antigens, such as influenza A viruses, are difficult to be controlled by vaccination. The viral genetics actually may help us to develop more effective vaccines. Some types of viral infections recur repeatedly, such as parainfluenza virus, or can persist for a long period of time in case of retroviruses, even in the presence of antibodies. These may be better controlled by antiviral drugs. The genetic analysis will help us to identify the virus-specific process that may be good target for the development of antiviral. So there is an important question. Why do viruses mutate? Simply, in order to survive. As viruses grow rapidly and produce large number of progeny variants per one cell, more chances of mutations may occur over a short period of time. However, the type of viral genome play an important role in their genetics. On the other hand, viruses must adapt or evolve the host immune responses, escape from them, by changing its surface proteins enough in order to trick the host cell and be able to attach to them. The genetic material inside the virus play an enormous role in how quickly a virus mutates. DNA viruses tend to be more genetically stable than RNA viruses because they seem to have an access to the proofreading as long as they use the same mechanism that the host uses in order to create their own DNA, which means that there are an correction mechanism in the host cell for the DNA repair. While the RNA viruses do not, they replicate without a similar proofreading activity, and as a result, errors of the genetic coding occur frequently. These errors that allow the RNA viruses such as influenza or HIV virus to mutate rapidly and to make it difficult for vaccines and natural immunities to keep up and repair or prepare for new strains of the virus. Genetic changes of the virus can occur by two principal mechanisms, either mutation or recombination. Alteration in the genetic material of a virus may lead to a genetic or a change in the function of the viral protein. Such changes may result in the creation of a new viral serotype or viruses with altered virulence. Mutation arises by one of three mechanisms, actually either by under the effect of physical mutagens such as UV light and X-ray, or by the natural behavior of the bases that will make up the nucleic acid, or through the failure of enzymes that are responsible for the nucleic acid replication. Types of mutations are either point mutation, one base will be replaced by the other, or by insertion of a new nitrogen base, or by deletion of one base. However, viruses differ markedly in their mutation rates, which is primarily due to differences in the fidelity of their enzymes that will replicate their nucleic. Mutation can produce viruses with new antigenic determinants. The appearance of an antigenically novel virus through mutation is called antigenic drift. New virus. Antigenically altered viruses may be able to cause disease in the previously resistant or immune host. I mean those hosts that have antibody to the first type of the virus. Mutation can produce viruses also with reduced pathogenicity, altered host range, or altered target cell specificity. The virus can target another cell in addition to the first or the original cell, which with also with intact antigenicity. They will retain antigenicity and differ pathogenicity. Such viruses can sometimes be used as vaccine strains are useful. Recombination involves in the exchange of genetic material between two related viruses during co-infection of the same host cell. Recombination by independent assortment can occur among viruses with segmented genomes only. Genes that reside on different pieces of nucleic acid are randomly assorted. This can result in the generation of virus with new antigenic determinants and new host ranges. Can infect another host or another type of host. Development of such viruses through independent assortment is called antigenic shift. We have to know the kinds of phenotypic changes seen in the virus mutations or during viral mutations or virus mutants. First of all, we have the conditional lethal mutants. These mutants multiply under some conditions but not others, whereas the wild type can grow under both sets of conditions. Example, the temperature-sensitive mutants. These will grow at low temperature, example 31 degrees centigrade, but not at 39 degrees centigrade, while the wild type can grow in both temperatures. It appears that the reason of that is the altered protein that cannot maintain the functional conformation at the elevated temperature. The host region mutants, these mutants will grow only in a subset of cells, of cell types, in which the wild type virus will grow. Such mutants provide a means to investigate the role of the host cell in viral infection. The plaque size mutants, plaque means the cytopathic effect that will, or destruction that the virus can produce in the infected cells. The plaque may be larger or smaller than in the wild type virus. Sometimes such mutants show the altered pathogenicity of the host. The drug-resistant mutants, another type of mutants, they are important in the development of the antiviral drugs. The possibility of the drug-resistant mutants arising must always be considered because virus as bacteria can produce a new drug. Another type of mutants are the enzyme deficient. Some viral enzymes are not always essential, so we can isolate viable enzyme deficient mutants. Example, the herpes simplex virus, thymidine kinase deficient, is usually not required in the tissue culture, but it is important in the infection of neuronal cells. Another type is the hot mutants. These can grow better at elevated temperature than the wild type virus. They may be more virulent since the host fever may have a little effect on such mutants. Attenuated mutants, many viral mutants cause much milder symptoms or no symptoms compared to the wild type or to the parent virus. These are said to be attenuated and have a potential role in the vaccine. So they are very useful. So what is the defective virus? Defective viruses, those viruses that lack one or more functional genes that are required for the viral replication. Many are undergo deletion mutants. So they need another helper virus in order to provide the missing function. or maybe steps in the replication or maturation of the virus. Some retroviruses have picked up the whole cell sequence but have lost some viral functions. These need a closely related virus which will retain the function of the defective virus as a helper. So some defective viruses can use unrelated viruses as a helper. For example, hepatitis delta, which is an RNA virus, does not code for its own envelope proteins, but will use the envelope of hepatitis B virus, which is a DNA virus, when these viruses are present at the same time in the same cell. Talking about the recombination is a process of exchanging genes between two chromosomes within regions of significant base sequence homology. This kind of break and join recombination is common in DNA viruses, or those RNA viruses with a DNA phase. The various uses for recombination techniques. It is useful for mapping of genomes, also for the marker rescue. What is the marker rescue? It is when the DNA fragment from a wild-type virus will recombine with a mutant virus in order to generate a wild-type virus. This provides a means to assign a gene function to a particular region of the genome, or will provide a means to insert a foreign material into a gene. Okay, the recombination also enable a virus to pick up genetic information from viruses of the same type, and occasionally from unrelated viruses or even the host genome, as occur in some retro. If a virus has a segmented genome, and when two variants of that virus infect a single cell, so the progeny recombinant variants will result from recombination of one genome fragment rather than an actual crossover event. This is an efficient process, but is limited to the viruses with segmented genomes. So far, the only human viruses characterized with segmented genomes are the RNA viruses, such as the Orthomyxoviridae family, reoviruses, arenaviruses, or Borneo-viruses. Reassortment is a non-classical kind of complementation. Complementation it is the functional interaction between two defective viruses, which permitting the replication under conditions inhibitory to a single virus. For example, two temperature-sensitive mutants infect the same cell. Each mutant can provide the missing function for the other in order to replicate. Nevertheless, the progeny virus will still contain the mutant genome and will be temperature sensitive. Multiplicity reactivation, it is if a double strand DNA virus are inactivated using by the ultraviolet irradiation, reactivation can occur at a very high multiplicity of infection. Lot of viruses per one cell at the same time. This is because the inactivated virus will cooperate with the other. The phenotypic mixing is a special type of complementation. It is the association of a genotype with a heterologous phenotype. It occurs when two different viruses infect a cell. The progeny viruses will contain a genome of one virus which become randomly incorporate within the capsid protein specified for the other virus, or a capsid consisting of a component of both viruses. One capsid protein and the component of two viruses. Phenotypic mixing may occur between either related or unrelated viruses. When occur between related viruses, different members, for example, of the Orthomyxoviridae family, or between genetically unrelated viruses, such as the rhabdo and the paramyxoviruses. We can also get this situation when a coat of other virus is entirely enveloped in another virus.